everyone, welcome to Crownedge. I'm Dr. Vijetha Vishwanath and I'm a restorative dentist and an endodontist. Today we'll be discussing about rationale and diagnostic aids in endodontics. Because it's a vast topic, we'll divide it into smaller topics into inflammation, role of immunity in endodontics, and systemic components, diagnostic aids, recent advances and miscellaneous. We'll discuss it into two parts. Part 1 is about rationale in endodontics. And this is inflammation and immunity. The most basic concept, the science of inflammation, they are the Celsius tetrad. The first one is, the 4 signs can be up whenever there's an injury or harmful thing inside the body. 4 signs can be appreciated initially. That is the redness, rubor. Second, warmth, that is calor. Third, swelling, tumor. And fourth, pain, that is dolor. So these 4 are the Celsius tetrad. And the 5th is the Virchow's functional Lisa. Now let us discuss about types of inflammation. Basically, there are 2 types of inflammation, that is acute and chronic, which is determined based on the duration. So in acute inflammation, the causative agents are pathogens, irritants, and whatever damage is there. And the duration is for a very short duration, like very few days, and the onset is almost always immediate. The most common question that is going to be asked are, what are the major cells that are found in acute inflammation? In acute inflammation, you have to note that the most common major cells that are seen in acute inflammation are polymorphonuclear neutrophils and macrophages. So whenever there's an acute inflammation, the outcome can be 3 things. Either it fights back and it leads to resolution of how it used to be before, or it can lead to abscess formation or because of accumulation of pus, or if it goes on for a longer duration, it can get converted into a chronic inflammation. So in chronic inflammation, it is basically because of persistent acute inflammation due to non-biodegradable pathogens or persistent foreign bodies. So it happens for duration is mainly up to for many months to years, and the onset is delayed. Again, the other question that can be asked from this particular sides are, what kind of what are the major cells that are seen in chronic inflammation? It is the monocytes, lymphocytes, macrophages, and plasma cells. So what is the end outcome of a chronic inflammation? It can lead to tissue destruction and fibrosis. So from this particular slide, the questions that can be asked are, what are the major cells that are seen in acute inflammation? What are the major cells seen in chronic inflammation? What is the onset and what is the duration and what is the final outcome? Now let us discuss about active immunity and passive immunity. Whenever there is any infection in our body, the end result from our side is the production of antibodies. So we can classify immunity based on how these antibodies are produced into active or passive. In active immunity, this first one is natural. This is whenever there's infection enters our body as a defense, it produces antibodies against them. The second type is artificial immunity, that is based on vaccination. Vaccination is basically they induce antibody production. So you can classify them into either when there's introduction of a whole agent vaccine or a subunit vaccine. In a whole agent vaccine, there are 2 types. When they are inactivated or heat-killed antibodies are heat-killed microorganisms are introduced into the body, like in rabies, or there is attenuated or weakened microorganisms which are introduced into the body, like MMR. And in subunit vaccines, they are basically just a part or a product of the microorganisms are introduced into your body. And the body develops antibodies against it, like in hepatitis B. The other type of immunity are the passive immunity. Passive natural immunity is what we receive from the mother. So the transfer of the maternal antibodies is into the fetus is called as passive natural immunity. Similarly, passive immunity, which has artificial immunity, in this especially in patients who are immunocompromised, we cannot introduce antigen into their body and expect them to produce antibody to fight against it. So in this, directly they will introduce monoclonal antibodies into the patient's body, and that fights against the infection. So types of immunity, again, there is innate immunity and adaptive immunity. Innate immunity, as the name says, it is something that is present since birth. And particularly, you need to know that they are not specific. They kill anything and everything that enters the body. They are not very specific about which particular microorganisms are involved. So in innate and the adaptive immunity, like you know, it is slow response. It is acquired over a period of time, and they are highly specific. So in this particular slide, the main type of questions that are going to be asked are, what are the types of cells that are seen in innate immunity? What cells? And the second question is whether they are specific or non-specific. So in innate immunity, they are highly specific. Their presence in birth, the cells which are responsible are macrophages, natural killer cells, dendritic cells, neutrophils, eosinophils, and basophils. Whereas adaptive immunity is slow response, and they are acquired over a period of time, and they are highly specific, and the cells which are involved are the B cells and the antibodies which are produced against that particular antigen, and the T cells. So let's start answering the questions based on what concept we have learned right now. So the first one is, which of the following convey the long-lasting immunity to an infectious agent? The options are naturally acquired active immunity, artificially acquired active immunity, naturally acquired passive immunity, artificially acquired passive immunity. By now we have already understood that whatever your body has produced actively, that is the best. So naturally acquired active immunity is considered something to be long-lasting immunity, because you have first-hand encounter with those antigens and your body can recognize them really well, and they will fight against it. Next, which of the following is a hallmark of acute inflammation? A. Polymorphonuclear neutrophils, agranulocytes, lymphocytes and plasma cells, and macrophages. We have learnt about it in the previous slide, about where we have learnt about the difference between acute and chronic inflammation. The most common question that is asked about acute inflammation is what type of cells are present, and it is always neutrophils. Similarly, so this is the answer. Now that we have learnt about neutrophils, for the same answer, two different questions can be asked, like which cell forms the front line defense in an innate front line of innate defense? Again, it is neutrophils. Second, which are the first cells to migrate from vessels to the site of inflammation? It is neutrophils again. So three things that we have learned. Neutrophils are the first line defense. They are the first cells to migrate from the vessels to the site of inflammation, and they are the main cells which are involved in acute inflammation. So we've learnt about neutrophils. So here, the polymorphonuclear neutrophils are attracted to the area of inflammation by what? So three options are complement system, enzymes, chemotactic factor, both A and C. Here we need to know that it is attracted to the site of inflammation by both the complement system and the chemotactic factor. So it is both A and C. How, now that we know that they get attracted to the site, we need to know how does it get attracted to the site. So any antibody or complement protein that enhances phagocytosis by marking an antigen is called what? It is more or less a definition. So the answer is opsonin. Opsonin is a substance which gets coated onto the antigen, which helps the neutrophils to identify it better. So for the same opsonin, what other question can be asked? So what helps in binding the bacteria to the surface of polymorphonuclear neutrophils? It is opsonin again. And if they ask you, a question is asked about what is the process called, like what is the name of the process by which the bacteria is made recognizable by polymorphonuclear neutrophils, it is called as antibody opsonization. So here we have learned three things. First is the bacteria gets attracted to the site because of the presence of opsonin. That coats over the opsonin is a chemical which is coated over to the bacteria, which helps the neutrophils to identify it better, and this process is called as antibody opsonization. Okay. So here, what cells act as a scavenger of dead cells, tissues, and foreign bodies? The options are again neutrophils, agranulocytes, lymphocytes, and macrophages. So here you need to be able to identify the scavenger, dead cells, tissues, foreign bodies. So scavenger is something that eats it up, and here in the options there's something which tells you clearly phages. Phages are something to eat it up. So whenever the polymorphonuclear neutrophils, they fail to remove the bacteria or fail to remove the bacteria, the circulating monocytes, they reach the site of inflammation and they change into macrophages. And so these macrophages are the ones which are responsible for phagocytosis and panocytosis. Let's talk about lymphocytes and the classification of lymphocytes. So basically lymphocytes are pluripotent hemopoietic stem cells. So based on what region they become immunologically competent, it is classified based on the location. So if they become immunologically competent in the thymus, they are called as T lymphocytes. Similarly, if they become immunologically competent in the bone marrow, it is called as B lymphocytes. So although there are many T lymphocytes and B lymphocytes, the most important four among T lymphocytes are memory T cells, helper T cells, suppressor T cells, and effector T cells. Similarly, in B lymphocytes, the two most important are the memory cells and the plasma cells. Now let us discuss about the T lymphocytes. So here the questions that can be asked about the lymphocytes are identify the which of the following are the T lymphocytes, which of the following are B lymphocytes, or where is the T lymphocytes, where do they become immunologically competent? It is there in the answer itself. T stands for thymus, B stands for bone marrow. So we'll talk about the four cells in T lymphocytes. First is the memory T cells. So in memory T cells, basically it speeds the immunological reaction in subsequent encounter with the same antigen. It is more like you've gone to a particular location, you meet somebody, and definitely it sticks into your memory. And next time when you meet the person, you know exactly who that person is. That's the same thing that works with the body. So there's an antigen which has infected your body, and these cells helps to identify them and they keep them in the memory. So subsequently, when the same antigen enters your body, immediately they'll be able to identify them. Second is the helper T cells. These are mainly stimulator. They are present in the acute phases of lesion expansion. So these are the ones which start off the inflammatory reaction, the helper T cells. They are present during the acute phases of lesion expansion. This is something you need to remember. Then the suppressor T cells. You can see the pictures just to remember it well. So the suppressor T cells are predominant in the later stages, preventing rapid expansion of the lesion. So obviously there's somebody who has stimulated it. If you just let it go on, it's just going to become bigger and bigger and bigger. Obviously you need somebody to stop it. So the main function of a suppressor T cell is to prevent it from expanding too much. So they are present during the later stages of inflammation. They prevent the rapid expansion of the lesion. These are the suppressor T cells. The fourth one is the effector T cells. They are the ones who interact with the host cells to carry out their immune function. They are responsible for cell-mediated immunity. This is most important. You will have to remember it because there are chances that you might get a question. which cell is which of the following cells are responsible for cell-mediated immunity. So it is T lymphocytes. If the option is lymphocytes, it's T lymphocytes. To be more specific, then it is the effector T cells. Now let us discuss about the B lymphocytes. They are the immunoglobulins. It is very easy to remember by, it is GAMED. You can just remember it as GAMED. Okay, the most number of questions are going to be asked about immunoglobulins. Most of it is covered in this table. So we need to understand about, if we can observe the IgG, E and D. Look at the type of structure that is present. They are called as monomer. IgG, IgE and IgD have monomeric structure, and IgA, you can see they are double, so they are dimer, and IgM is a pentamer. If any question is going to be asked about the structure, this is what is going to be asked. So after we learned about the questions that can be asked about the structures of immunoglobulins, the next most common questions that are going to be asked about the features of each. So IgG, they are the most abundant immunoglobulins present in the body. They make up to 80% of the total content in the body. Secondly, they are the ones who are responsible for oxinization, and they are the only immunoglobulins which crosses the placenta from the mother, that is from the mother to the fetus. Second, IgA, they are the secretory immunoglobulins, and they mark the first line of defense against the infection by microorganism. They make up to 13%. So IgA are secretory immunoglobulins. Next comes IgM. They make up to 6% of the total, and they are the first antigen which are produced in primary response to an antigen. These are the first ones which are, although IgA is the first line of defense, IgM are the first antibodies which are produced against any antigen. Then these are the first antibodies produced by the fetus. You need to remember not to get confused. The IgG are the immunoglobulins which crosses the placenta from the mother to the fetus. But the first immunoglobulins which are produced by the fetus are IgM. Then IgE, they are less, 0.002%. They are associated with hypersensitivity reaction, and they have defense against they are the ones which are responsible for defense against the parasitic infection. So remember hypersensitivity and parasites. IgD, they make up to 1%, and they are membrane-bound immunoglobulins on the surface of mature B cells. The exact functions of IgD is not clearly explained. So the questions that can be asked from this particular table are based on the structure and based on the function and based on the total amount or the content present in the body. Most commonly they will ask which one is the maximum, which one is the most abundant immunoglobulin which is present, that is 80% IgG. So let's answer questions based on the lymphocytes and cell-mediated immunity. Cell-mediated immune response involves the destruction of infected cells by which of these? Basophils, T lymphocytes, B lymphocytes, and mast cells. The answer is T lymphocytes. To be very specific, it's the effector T cells. Like we had learned, they are the ones who are responsible for cell-mediated immunity. I had marked it in the previous slide for you to remember it better. Next, which of the following cells appear during the later stages of apical periodontitis? So the key word over here is later stages. Now let us discuss the options. There are 4 options: the memory T cells, helper T cells, suppressor T cells, and effector T cells. So just to revise it, memory T cells are the ones which are responsible for identification of the antigen when they come back again. Helper T cells are the ones which are responsible for during the initial stages of any infection. They are the ones who start off with the inflammatory response. Suppressor T cells are the ones which come during the later phases. They are mainly to suppress the expansion or prevent rapid expansion of the lesion. And fourth is the effector T cells. Just the previous question we learned that they are responsible for cell-mediated immunity. So the answer is a suppressor T cells. Cells which are seen during the later stages are suppressor T cells. Early stages are helper T cells. Next question. Humoral immunity is mediated by basophils, T lymphocytes, B lymphocytes, or mast cells. Immediately you need to think about the immunity that we had learnt about innate immunity and adaptive immunity and the Venn diagram we had learnt. So the adaptive immunity is something which comes over a period of time, and those are the B lymphocytes are the answers. They are highly specific, and they have slow the three things that we had learned. They have slow response, they are highly specific, and they are acquired over a period of time. Next, synthesis of antibodies takes place by which of the following cells? Lymph, T cells, B cells, and bone marrow. So antibodies are also called as immunoglobulins. So we just learned about the 5 immunoglobulins, and we came under the heading B lymphocytes. So that gives the answer, that is the B cells. B cells are responsible for immunoglobulins. Now let us discuss questions about immunoglobulins, whatever we've learned in the table. So first one, which is the most abundant immunoglobulin in the body? The thing to concentrate is the most abundant immunoglobulin. We have learned it. It is IgG. IgG makes up to 80% of the total immunoglobulins. So the same answer for immunoglobulin G. What are the questions can be asked? They can ask about which of the following antibody directly takes part in opsonization process. The keyword is opsonization. Or they can ask which immunoglobulin passes through the placenta. So 3 things that you can be asked about IgG is which is the most abundant. Second, opsonization. And third is which crosses the placenta.